Hello everyone, I am Ruchira and welcome to my channel. I am going to start an education series on Basic and Applied Microbiology, and this is the first video. Before we begin, I want to start with what is microbiology. We all know that doctors study human body, veterinary doctors they study non-human body, mechanical engineers they study machines. Similarly, microbiology is the study of microbes. Now, what are microbes? We simply say that microbes are those organisms which are not visible through our naked eye. Due to this COVID situation, even people belonging from non-biology background, even they know what are microbes. But the question is, how did we came to know about the existence of microbes? If they are not visible through our naked eye, then how we came to know about microbes? So this episode is all about discovery of microbes or history of microbiology. So let's proceed with the video. In history of microbiology, two important points that we should remember is spontaneous generation theory and golden age of microbiology. Though spontaneous generation theory is a hypothetical concept, but it is a very important milestone in the history of microbiology because several scientists opposed to this spontaneous generation theory, and that gave rise to the golden age of microbiology. So before studying golden age of microbiology, we should definitely study what is the spontaneous generation theory. So we will start with this spontaneous generation theory. In the very early ancient times, there were some theological and mythological explanations to existence of life. But from 600 to 500 BC, some Greek philosophers proposed that living organisms spontaneously originated from inanimate objects. During this period, Anaximander proposed that fish or fish-like animals emerged from warmed-up water and earth. Not only that, men and women came out from these fish or fish-like animals. In 300 BC, Aristotle proposed that invertebrates like mollusks, arthropods, they emerged from mud. There were also concepts like if we keep cheese or bread in rag and leave it in a dark corner, mice will emerge. Even there were concepts like decaying meat gives rise to maggots, that is, larvae of flies. Thus, this theory was also known as abiogenesis, which means that living organisms originated from non-living objects. These hypothetical concepts surfaced for almost 2000 years, but finally in the 17th century, Franciscoreddy opposed to this concept by performing some experiments. In his experiment, he took 3 containers. Container 1 was kept uncovered, container 2 was sealed with a paper cover, and container 3 was covered with a fine gauze so that flies cannot enter the container. What he observed was quite surprising. In the container number 1, which was uncovered, flies entered the container. They laid eggs on the meat and maggots were developed. Coming to container 2 that was covered with paper, no flies entered into that container and no maggots were formed. Container 3 that was covered with fine gauze material, no flies entered the container and no maggots were formed inside the container. However, the flies were attracted towards the fine gauze material somehow, and they laid eggs on the surface of the fine gauze where maggots were formed. From this experiment, it was inferred that maggots developed from fly eggs. It did not develop spontaneously from decaying meat, but whenever the meat was contaminated with fly eggs, maggots were developed. Even when the gauze covering the container 3 was contaminated with fly eggs, maggots were developed. Thus, this experiment opposed the concept of spontaneous generation, which said that living organisms originated from non-living things. In a similar time frame, Antony van Leeuwenhoek also gave some descriptions about microorganisms by using his own microscope. Actually, he was a draper, that is, a kind of retailer of clothes. For running his draper's shop, he wanted to visualize the quality of the thread better than a magnifying glass. So, in this process, he developed some curiosity in making lenses, and he constructed simple microscopes that could magnify objects from 50 to 300 times. So, in this process, using his own microscope, he observed some tiny organisms and he drew them, and then he sent the description to Royal Society of London. His drawings clearly indicated bacteria and protozoa. So, these findings again conflicted with the spontaneous generation theory. After all these conflicts, there were some changes in the spontaneous generation theory. People believing this theory, they proposed that larger organisms did not emerge by spontaneous generation, but microorganisms rose by spontaneous generation. They also exampled that boiling extract of meat or boiling extract of hay, when it is allowed to settle down for some time, give rise to microorganisms. Even after the experiments of Franciscoreddy and Antony van Leeuwenhoek, the commotion regarding spontaneous generation theory was too high. People who favored spontaneous generation theory, they said that when meat is boiled in a broth and the container is tightly closed, it produces a cloudy atmosphere that contained microorganisms. But some people also tried to oppose these concepts by saying that no, only when the meat broth is exposed to air, it is getting the microorganism. So it is possible that air contains microorganism, and when this meat broth is getting contaminated with air, it is producing microorganisms. However, people who strongly favored spontaneous generation, they disregarded this fact and said that microorganisms can grow even without air contamination. Finally, Louis Pasteur stopped all these controversies through his path-breaking experiment. By 1861, Louis Pasteur settled the controversy on spontaneous generation theory. What he did was, first he filtered air using a cotton, and he found that some plant spore-like objects in this cotton. And then he sterilized a liquid medium and placed the same cotton that has been used to filter air in the sterile medium. And yes, he found microbial growth did occur in the sterile medium which contained the cotton. In the next experiment, he took a round bottom flask with long neck. He poured liquid medium in this flask. Next, he heated the long neck to give it a curved shape like swan's neck, and then he kept the mouth of the neck open so that the flask is exposed to the atmosphere. Next, he boiled the liquid inside it for few minutes and allowed it to cool down. Surprisingly, he found no microbial growth in the liquid medium, but when the curved neck was broken, microbial growth occurred in liquid medium. This experiment is also known as Swan neck flask experiment, and it is a path-breaking experiment in microbiology. Louis Pasteur clearly pointed out that it is air that carries microorganisms. When a sterile liquid medium comes in contact with air, it actually gets exposed to microorganisms. In the Swan neck flask experiment, the curved neck prevented microorganisms from entering into the flask. The microorganisms got trapped into the curved neck and the liquid medium remained sterile. But when the neck was broken, the medium got exposed to air and it got contaminated with microorganisms. He completely disapproved the theory of spontaneous generation of microorganisms. Finally, in 1877, John Tyndall gave a final blow to the spontaneous generation theory. He demonstrated that in air, dust particles actually carried germs. If the sterile broth comes in contact with air that does not contain dust particle, the medium will remain sterile. And this was the end of spontaneous generation theory, and also it marked the beginning of golden age of microbiology that will be discussed by me in the next video. For any questions, please feel free to ask me in the comment section, and I hope this video is quite informative, and so please like my video and also share with your friends, and don't forget to subscribe my channel to get more videos on basic microbiology and applied microbiology.